Well, welcome everybody to this October BMS Talk. I think we'll get started now and I'll introduce our chair. Chris Knowles is a field mycologist who carries out fungus surveys and monitoring at sites across the UK and is an active member of the Edinburgh and Lothians Fungus Group. Chris is an associate tutor with the Field Studies Council and works in the herbarium at the Royal Botanic Garden, Edinburgh. So thank you very much for being the chair, Chris, and I'll hand over to you.
All right, thanks, Sally. It's a pleasure to be here. Before I begin, I'd like to invite all of you to find the chat icon on your screen. It's usually at the bottom, and you sometimes need to wiggle your mouse down there a bit, because then at any point during the talk, if you can pop any questions that you have or think of during the talk, pop them into the chat. I'll be noting them down and I can bring them up at the end of the session for a Q&A that we'll be doing. On which note, I'm very happy to introduce Dr. Chris Ellis this evening. Chris has a long history working at the forefront of climate ecology and lichen research and has had fantastic opportunities to be working with other esteemed lichenologists along the way. After completing a degree in ecology at the University of Leeds, Chris went on to complete a PhD in paleoecology at the University of Manchester, where he was studying the effects of past climate change and human land use on British uplands. From there, Chris first became interested in lichens following a move to Canada while he was investigating the sensitivity of high Arctic environments to past and future climate change. On returning to the UK, he took up a position in Peter Critterden's lab at the University of Nottingham, studying the nutrient uptake of mat-form lichens. Chris then moved in 2003 to the Royal Botanic Gardens in Edinburgh, working initially with Brian Coppins and then going on to focus on patterns of lichen diversity, including their responses to threats, including climate change. And he's still there to this day, currently working as the head of cryptogamic plants and fungi and co-leading RBG's research section on conservation science. So with that very brief summary of Chris's many achievements, I'll now hand over to him for his talk this evening.
Okay, thank you very much, Chris. And thank you everybody for the opportunity to give this talk this evening. It's a real honor to have been invited to talk to the BMS. So I'm going to jump straight in. The structure of the talk, I was asked to explain a little bit about lichens. So there's a quick introduction to lichens. Then there's a quick introduction to climate change. And then I'm just going to explore briefly why this intersection of lichenology and climate change matters. And I'm going to move on to think about how we typically study climate change and the impacts of climate change with one kind of baseline study, if you like. And then I'm going to present 4 pieces of research, 4 interrogative studies that kind of unpack that typical approach based on what we know about the biology of lichens. So how studying lichens can improve, if you like, our understanding of climate change and climate change impacts. And then I'll finish with some conclusions. So first of all, a quick intro to lichens. So we're talking about fungi principally, and just to go to the basics, fungi don't photosynthesize, they're heterotrophic. So we know that fungi need to absorb a source of food from outside their own tissues, and they do this in a variety of ways. They might form an association with plant roots as mycorrhiza, they might decompose organic matter, or they may exist as parasites. And lichens are fungi that have evolved a very particular mode of nutrition. So they build this lichen structure, this thallus. Most of the biomass of that structure is fungal material. And if we take a slice through that thallus, slicing a carrot, here we are slicing through a lichen, then we can see certain structures. So we can see an upper protective cortex, an outer layer. We can see a population of algae sitting inside the lichen body. We can see a medulla composed of fungal hyphae, and then in this case, a lower cortex, a lower protective structure. So lichens have fungi, lichenized fungi. They've evolved to generate these structures within which they can contain a photosynthetic partner, and either an algae or a photosynthetic cyanobacteria. And here's an electron micrograph that shows the photosynthetic cell, the algal cell, surrounded by fungal cells. So that algal cell is photosynthesizing. The fungus induces a leakiness in the cell membrane and can absorb some of the sugars, the photosynthetic product. But the fungus, in building this structure, also protects the algae. So it protects it, for example, from herbivores or from the impacts of UV light or whatever that might be. So this symbiotic relationship. So most of the fungi that have evolved this mode of nutrition, this lichenization lifestyle, occur within the Ascomycetes. So in sister to the Basidiomycetes and within the Pezizomycetina clade within the Ascomycetes. So 98% of lichens are Ascomycetes. They have these, their spores are produced in these little flasks, these little asci. There are an estimated 20 to 30,000 species of lichen fungi globally. And at a recent count, that represents about 25% of that Pezizomycetina clade. And there's about 2,000 species of lichen fungi in the UK. So when we're thinking about the taxonomy of lichens, when we're referring to different lichen species, of course, we're referring to different fungal species. So we've got cross sections of different types of lichen here. You can see the upper cortex again, lower cortex. In this case, we've got little packages of cyanobacteria. And here you can clearly see the fungal hyphae and chains of cyanobacteria encased within that thallus. So different lichen species are different fungal species, and those different lichen species may associate with the same or with different algae or with different cyanobacteria, depending on patterns of coevolution. But not all the lichen fungi don't form a single group. So that process of being a lichen, that mode of nutrition, appears to have evolved independently multiple times within the sort of fungal tree of life, if you like. So here's an early study from the 90s that demonstrates that there are a few Basidiomycetes that form lichenized structures, but two independently evolved clades here of lichen fungi, and then within these clades, this is another phylogeny showing relationships among lichen fungal species. Within these clades, this study suggested that there was one single origin of lichenization and then a loss of lichenization multiple times. So these really complicated nested structures that explain how that process of lichenization has evolved within the Ascomycetes. And that's as far as I'm going to go, thinking about the evolution of lichens, and there are a lot of interesting advances on this early work. And I think if you wanted to explore that further in the BMS, someone like Esther Geyer from Kew or my colleague Rebecca Yar from RBGE would be able to talk about evolution of lichenization and coevolution with the algae or cyanobacteria. What's important to me as an ecologist is the incredible diversity of phenotypes that have evolved. So the shapes and the sizes and the colors and the anatomies of lichens, and trying to understand how that diversity of form fits individual species, their adaptations that fit them into particular environments and microenvironments, and how we might use that diversity to ask questions and to learn about the effects of environmental change, including climate change. So an important point at this stage of the presentation is also to think about how lichens are poikilohydric. So that means that they're unable to regulate their water content. They don't have xylem and phloem and roots and stomata and so on. So if the environment is dry, then the lichens are dry, and if the environment is wet, then the lichens are wet. And that's got really important consequences for their physiology. And we can see that here in this graph, where we've got net photosynthesis or net carbon uptake in relation to the water content of the lichen thallus, the lichen body. And you can see that as the water content increases, initially we get respiration, and respiration continues, so negative carbon loss. But then photosynthesis kicks in, and photosynthesis rises as the thallus gets wetter up to a peak and optimum. And then as the thallus gets saturated, because of the slower diffusion of gases, oxygen and carbon dioxide across water films, photosynthesis in the green line starts to fall again. So you get an optimum, and then you get this process of supersaturation where photosynthesis declines. And the type of photosynthetic partner is really important in understanding these kinds of hydration dynamics. So here's an example with a green algal lichen, and again we've got carbon dioxide exchange on the y-axis, and in this case we've got time on this x-axis, and this green algal lichen has been introduced into a humid environment, and just in a humid environment with water vapor, it can start to photosynthesize straight away. So we've got this positive carbon exchange. And then as it gets supersaturated, this is a point with the arrow where liquid water is added, then photosynthesis declines. But for cyanobacterial lichens in the same experiment, when you introduce a dry lichen into a humid environment with water vapor, nothing happens until the point when you add liquid water. So these cyanobacterial lichens, lichen fungi associated with cyanobacteria as their photosynthetic partner, depend on liquid water for activation. So a big difference in how green algal lichens or cyanobacterial lichens interact with the environment. One of the recent exciting things in lichen ecology is the way that we've started to develop metrics, a bit like our vascular plant colleagues have got things like specific leaf area that measures the, it's an important metric measuring measuring the adaptation of leaves to particular environments. A series of metrics have been developed. So the first one is referred to as specific thallus mass, which is the milligrams of dry matter per centimeter squared. The water content is simply the milligrams of water per milligram of dry mass. And then the water holding capacity is the milligrams of water that a lichen can hold per centimeters squared. And we see lichens with these different values for these metrics, falling into different places along a gradient of wetness and dryness. So species that have got a high water holding capacity, they need more water to reach that optimum point of wetness where photosynthesis is maximized, but they're less likely to suffer supersaturation. So they can absorb a lot more water before those water films start to develop. And they're often associated with cyanobacteria and occurring in wetter environments. Species with these lower water holding capacities, they need less water to reach optimum photosynthesis, but they're more likely to reach supersaturation if the environment is very, very wet. And they tend to be associated with green algae that can start to become active and photosynthesize just using water vapor, so occurring in drier environments. And recently, some exciting work by Ingvar Gausler's group in Norway is thinking about the way that you might measure lichens using these different metrics and position them in with respect to the availability of moisture and also the different types of moisture. Has the environment, is it dominated by a moisture that's available from humid air or rainfall or dew? And you can see lichens falling out into these different places as these morphologies represent adaptations to the uptake of different moisture types and different amounts of moisture. So a little bit of an introduction to lichens and lichen ecology. Very briefly about climate change. I thought I'd show this graph. So on the x-axis, we've got time in initially millions of years, from 60 million to 10 million, 9 million to 1 year, 1 million, and then thousands of years, and then recent time with climate projections into the future. And over those time periods, we can see atmospheric carbon dioxide concentrations falling over time with our glacial and our interglacial cycles, and then projections of future carbon dioxide concentrations increasing because of human activity, and then mapping that to changes in global temperature, just to emphasize the scale of the problem and the awesome scale of climate change. So through to 2300, which sounds a long time away, but in the life of a woodland, for example, isn't that far at all. We're looking at temperature increases in a business as usual scenario, that, you know, temperatures that haven't been seen for perhaps 50 million years. So huge problems, and problems that are recognized in global reports that are available. This is a report that's worth looking at, the Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services, which summarizes the state of biodiversity and the drivers of biodiversity change, and suggests many species are unable to cope locally with the rapid pace of climate change, and climate change is projected to greatly increase the number of species under threat. And that includes lichen species. So we've looked at lichens, and we've thought a little bit about lichens, and we're all familiar with climate change. And we've thought a little bit about climate change, and now we want to bring those two things together. But I think, first of all, I guess I don't need to do this with this audience. I think, you know, I'm preaching to the converted, but it's always worth thinking, you know, those challenging questions about, given the scale of this problem, you know, why does it matter? Why should we be worrying about what the consequences of climate change are for lichens? And I do like to sort of revisit these questions and to hear what other people have to say about this. But the first obvious point is the ethical one, perhaps less popular these days, but stewardship of the environment, including biodiversity. The point that's more often made these days is this sort of enlightened self-interest, because they're functionally important. So changing lichen distribution, changing lichen biomass, changing the functional composition of lichen communities, all that stuff has got consequences. And here's a nice figure from a paper that was published a few years ago in New Phytologist that looks at a latitudinal gradient, and it thinks about what are the main functions that lichens and bryophytes and algae, these cryptogamic covers, what are the main functions that they're performing along this gradient, from having an albedo effect in the tundra or the northern forests that creates surface cooling, carbon sequestration in our peatlands, nutrient cycling in forests. Lichens are really, really good at absorbing nutrients from the atmosphere, protecting soils in dry lands, and partitioning precipitation in tropical forests, and so on. So there's a lot of evidence now to suggest that lichens have these very important functional properties, and that it's of consequence for ecosystem function and services. And what's not on this graph is that includes these trophic cascades. So here's a study that was published that looked at lichen biomass on twigs, so epiphytic lichens growing on twigs, and oh, that's in the way, I can't see. The number of invertebrates on the y-axis. And you can see that as lichen biomass goes up, so does the number of invertebrates. So lichens are creating a habitat for these small invertebrate species across a range of different taxonomic groups. And subsequent studies have shown that that has consequences for bird populations, for example. So lichens playing this role in ecosystem food webs. I think one point that's often overlooked, though, is the taxonomic bias. So we know there's a taxonomic bias in science, and certain groups of organisms tend to be preferentially studied, and there are an increasing number of papers that demonstrate this now. But here's a nice paper that looks at the relationship between the amount of biodiversity data that we've got for different taxonomic groups and the effort that goes into collecting that data, and then the species richness of those groups. And in green are the positive residuals, where we've got more biodiversity data than we'd expect, given the species richness of those groups. And the red are the negative residuals, where we've got less biodiversity data than we would expect, given the species richness of those groups. So the greatest positive residual is, of course, the birds. We've got loads and loads of data about birds, biodiversity data about birds, distribution data relative to their species richness. And just picking out two fungal groups here, you can see that for these fungal groups, we've got less biodiversity data than is justified by their species richness. And I mean, it's pretty profound, really, because it forces you to ask, what kind of knowledge and what kind of learning are we actually missing through that kind of bias? Our science, in a sense, is a bit unscientific because we're not concentrating our efforts where the species richness is. We're concentrating our efforts on the things that people tend to like. And there's a really serious gap. Could our environmental management, could our response to issues like climate change be more consequential and more effective if we more truly represented biodiversity on planet Earth, if our science was more representative of the biodiversity that exists? Okay, so that's what I want to try and exemplify through a series of studies that look at lichens and climate change, and in their own small way provide some, reveal new information, new ways of thinking about how we approach climate change. But before I can get onto those studies, I just want to demonstrate how a lot of climate change research is done. So a typical way of conducting an analysis on species risk. And bioclimatic modeling is a very popular way in which we think about the risk to species. And there's thousands and thousands of studies that use bioclimatic modeling to look at climate change impacts. So what we would do in a bioclimatic modeling study is we would have some measure of the fitness of the species on the y-axis. It's growth, physiological tolerance, occurrence, abundance, whatever that might be, compared to a gradient, an environmental gradient, often temperature, on the x-axis. And we build a niche model. So the performance of the species would likely go up to an optimum and then decrease. And if the 2024 temperature, I'm in Edinburgh, so if the 2024 temperature in Edinburgh was the species optimum, then we can think about how that temperature might change in the future based on climate scenarios. It's going to get warmer. So it will shift along this axis to a different point, and we can characterize the impact on the species as the change along the y-axis. So we've got a 2024 temperature, we've got a scenario temperature in the future, and using this niche model, we can look at the impact on the species. So this is how a lot of research is performed. You've got a distribution map, which is the response. You've got a series of climate layers that your explanatory variables. You build these niche models, and then you project them back onto the climate layers to look at patterns in likelihood and the probability of occurrence. And then you do that for a baseline climate, and you do it under a set of climate change scenarios where the temperature is different. So here's an example for some Arctic alpine species in Britain, where the areas in red show areas of high likelihood of occurrence, very suitable climate in the present day through to the 2050s. And you can see that contraction in suitable climate space, which is presents a measure of risk to that species. So what you're doing is you're building a picture, an envelope, a spatial region of suitable climate space. and you are then using climate scenarios to characterize the change in that climate space, either its contraction or maybe a spatial shift in climate space, and then measuring the expected risk to a species based on those models. There are several problems with that approach. So one of them is that the assumption is that the sample distribution is in equilibrium with the climate. So the sample distribution represents the species' direct response to the climate. And one thing it doesn't include, for example, is any explicit information about species interactions. The second problem is that the kind of climate data that we use is incredibly powerful, and you know, there are fantastic sets of data, climate change scenarios that we can use to understand future climates, but they tend to include a set of canonical variables. So mean temperature, annual mean temperature, monthly mean temperature, precipitation totals for the year or for a season, and then there's spatial resolution. Even at very high spatial resolutions, it's in the region of, say, 10-kilometer grid squares or 1-kilometer grid squares, and you can pack a lot of climatic variation, microclimatic variation, into a 1-kilometer grid square, especially if you're in the mountains. So there's a set of issues to be dealt with. And we're going to explore those now in my 4 small research examples. So 4 studies, and each of them deals with one or more of these issues in our typical approach to climate modeling. This direct response to climate, this use of these cardinal variables, mean temperature, total precipitation, and then thinking about spatial scale. Okay, so my first example relates to lichens in mountain environments. And in this case, we sampled a series of plots focusing on mat-forming lichens. So these are lichens that tend to grow vertically, often quite spiky, holding themselves together in mats or intercalated amongst the wider vegetation. And we surveyed these lichens along altitudinal transects, multiple altitudinal transects in the Cairngorms. So Cladonia portentosa here, Flavostrira nivalis, Alectoria species, Alectoria oculuca, Alectoria sarmentosa will occur in these kinds of habitats. And we sampled the vascular plants as well, and along the altitudinal gradient there were 4 different vascular plant communities that we could identify, which are listed here, and they're shown here on this ordination plot, where the circles, the size of the circles shows the altitude from low altitude to high altitude. So you can see the separation of these vascular plant communities along this altitudinal gradient. The canopy height of the vascular plant communities goes down as you go up the mountains, and the amount of bare ground increases. But we were also interested, we were inspired, this is work that was done by an MSc student. We were inspired by this paper from 1947 by Watt that described the effect of wind on this type of vegetation. Very, very high wind speeds that structures the vegetation and maintains a low canopy. So we're also interested in the effect of wind speed here. So we found that as you increase in altitude along this x-axis, the canopy height of the ericaceous layer goes down. So the canopy gets smaller and smaller and smaller. The amount of bare ground goes up, and the abundance of lichens, the occurrence of lichens, peaks at the intersection of those 2 trends. So you get a kind of mid-altitude peak in the abundance of lichens. So at the low altitudes, the lichens are being outcompeted by this tall growth of Calluna and Erica. At the higher altitudes, the amount of bare ground, there's very little for the lichens to establish into, to hold on to these spiky growth forms, and they can easily be blown away. So you've got a stressful end of the gradient, and you've got a competitive end of the gradient. And at a point before you reach the mountain summits, when the vascular plant canopy is about 5 centimeters high, that's when you get the greatest colonization. So we were interested in the effect of altitude, temperature, and wind speed on structuring those lichen covers. So we built a little model, a thing called a path model, where you can look at the effect of altitude on canopy and the effect of canopy on lichen cover, or the effect of altitude and temperature on lichen cover directly, and the same for wind speed. So you can map out the different relationships, whether they're direct or indirect. And if we use that model and we think about what happens if we increase the temperature in the mountains, then just focused on temperature, the lichen occurrence goes down at lower altitudes and it would increase at higher altitudes. So you've got a shifting zonation of where the lichen cover is occurring. But then if we incorporate wind speed too, if we lower the wind speeds, then that tends to exaggerate the effect of temperature. But if we increase the wind speeds, then that tends to offset the effect of temperature and maintains a low vegetation cover. And a percent change in wind speed of about 15%, based on this model, a 15% increase in wind speed would be enough to maintain the cover of those lichens, given our expectations of temperature change, temperature warming. So what we've done here is thinking about the lichens, thinking about vegetation structure, thinking about lichen ecology. We've incorporated biotic interactions. We're thinking about competition and facilitation and where these lichens are facilitated within the vegetation, how that captures an indirect climatic response in response to a different variable, not temperature, not precipitation, but wind speed. And the challenge there is that our confidence in changes in wind speed in the future is very, very low. But introducing a new and challenging variable into our understanding of climate change effects. Okay, so continuing with a similar theme, but this time moving to epiphytes growing in temperate rainforest. So growing in the UK's temperate rainforest environment along the west coast of the UK. So studying in this case a particular species, Flavopalmelia caparata. Now we've done some bioclimatic modeling on this species in the past, and the bioclimatic models suggested that if the temperature gets warmer, then this species will benefit. So areas of orange and red in these graphs show areas of suitable climate for the baseline for today's climate in the 2050s and the 2080s. And we can see an improving situation for this lichen. It's a lichen that tends to grow in warmer places and likes warmer temperatures. So some temperature warming might be favorable. But what we did was we brought in some knowledge of the physiology of these species, and in particular something called the I-wet hypothesis. So these graphs here show carbon dioxide exchange, photosynthesis, respiration for wet lichens, but for different levels of light, so different levels of irradiance. And we can see that when irradiance is very low, you've not met the light compensation point for photosynthesis at low light, but the lichens are wet and active, so they're respiring. So there's more respiration. And then as the irradiance goes up, you get photosynthesis. So the hypothesis is that growth is driven by the amount of light the lichens receive when they're wet and active, and the amount of light is critical. Now, if we look at the climate change scenarios for the UK, we can focus on temperature on these left-hand panels. And this study was done in Scotland, so I'm going to highlight the Scottish bars here. And we expect summer temperatures to go up by 2 to 3 degrees, and we expect winter temperatures to go up by 2 to 3 degrees. So summer and winter temperatures, under a set of climate change scenarios, we expect them to go up. If we look at precipitation, we expect precipitation to go down in the summer So summer precipitation declining and winter precipitation increasing. So there's a seasonal difference in the pattern of precipitation that is expected under climate change. Lower summer precipitation, higher winter precipitation. But the amount of precipitation, the total amount of precipitation, can stay the same. So under a baseline, if we have an environment now with an annual total precipitation of 1,500 millimeters a year, it's wetter in the winters and it's a bit drier in the summers. Those are the sort of baseline climates. But we're increasingly seeing, and the projections would suggest that we have these wetter winters, but drier summers, and so a seasonal difference in precipitation, even though the annual total stays about the same. Now, if we take that eye wet hypothesis and we think about the impact of light, what we end up with then is lichens being very, very wet or increasing in wetness at a time in the winter when there's very low light, and being very dry and potentially dormant at a time in the summer when there's high light. So although the total amount of rainfall doesn't change, because of those seasonal differences and the interaction with light, the consequences of that seasonal shift could be pretty important. So we've got this kind of non-analog situation where the lichens could be wetter during a low light season, forcing higher respiration, and they could be drier during a high light season with lower photosynthesis because they're dormant. So we looked at Flavopalmelia caperata at a particular site in southwest Scotland. And so here are our bioclimatic models, and we can see that at this site we expect the environmental suitability for this species to go up from about 50% probability of occurrence to 80% probability of occurrence in the 2080s because of that warming. And at the site, we characterize the winter moisture, the winter temperature, and the winter light conditions that the species were exposed to, the individuals of the species. And then in the summer, the moisture, the temperature, and the light conditions that the individuals were exposed to in the summer. And then we grew the lichens under a set of simulated conditions. So for the baseline climate, using winter moisture, temperature, and light, summer moisture, temperature, and light, and then for a 2080s climate, using the 2080s winter moisture, temperature, and light, and the 2080s summer moisture, temperature, and light. And what we found was that if we took the winter baseline temperature here, then the growth rate was pretty neutral. The lichens under the current climate don't appear to grow very much. But under the summer baseline, summer conditions, the lichens appear to grow. So they're sufficiently wet under a high light environment in the summer, and you've got growth. But if we go to the 2080s scenario, then in the winter, when they're more wet, under the dark conditions, under the low light conditions, then you've got respiration and you've got this negative growth. And also in the summer, you have negative growth. So the net outcome for the lichens was that under those baseline conditions, they could grow. And under the 2080s environment, when you include that light interaction, you get mass loss, you've got negative growth. So taking into account that relationship with light completely overturns the expectation from standard bioclimatic models. And suddenly, instead of thinking the future might get better because of increased warmth, you've got this shift in seasonal precipitation, you've got the importance of that wetness for lichens and lichen biology, and you've got its relationship with light and a completely different outcome. So we continue to explore those kinds of relationships, particularly the relationship between lichen growth and hydration, different moisture sources, the amount of light, in a series of simple growth experiments where you can attach lichens to fishing line using a little bit of non-toxic silicone, and you can grow them at different sites, different climates, And then using clips that measure electrical conductivity, you can attach those to the lichens. It's very simple stuff. You can measure the conductivity and you can calibrate that with how wet the lichens are. So when they're wet, they conduct more electricity than when they're dry. And so you can use these little impedance clips to monitor in real time how wet and dry the lichens are. And so we did this for another temperate rainforest lichen. It's widely occurring, but it's very common in temperate rainforest environments, Liberia pulmonaria. And we can see for different months of the year that in this case, we compared the wetness of the lichens to the vapor pressure deficit, which is the amount of the drying effects of the air, if you like, how much evaporative force the air has got. And the open symbols show the vapor pressure deficit, which, for example, in June, this is the early morning. It's low in the early morning. It rises to a midday peak and then drops. And then this is the lichen wetness. And we can see that lichens are wet and then they dry out as the vapor pressure deficit goes up and then they get wet again. And we have this really strong coupling between the lichen hydration and physiological activity and the pattern in the vapor pressure deficit. So a really strong coupling. And this is for a site that gets about 3,000 millimeters of rainfall a year. But when we look at the water content of the lichens and we compare that to the rainfall, the relationship's quite weak, if there's a relationship at all. But if we look at the water content of the lichens and we compare it to the vapor pressure deficit, then we have this really, really strong relationship. And that drives the lichen growth. So we can see how the lichen growth is related to the amount of time that they spend hydrated in the light, driving positive growth, and the amount of time they spend hydrated in the dark, which where growth becomes less because you've got more respiration than photosynthesis. So what we're doing now is we're downscaling. We're trying to downscale this to think about climates at much smaller scales. So remember the climate data that's typically available is at a 10-kilometer grid scale or a 1-kilometer grid scale. Well, for a series of national nature reserves, we're taking the boundaries of the reserves. We're thinking about those large-scale climates, but then also small-scale patterns in altitude, in heat load, the amount of heat that a surface receives, maybe to do with the slope and the aspect, topographic wetness, whether it's a water shedding site or a water gathering site, canopy cover, and we're downscaling the vapor pressure deficit to a 5-meter scale. So to understand very, very high resolution changes in the climate variables that lichens are responsive to. So what we want to do eventually is convert that downscaled vapor pressure deficit, that 5-meter scale, to lichen growth and performance, species occurrence, species abundance, community structure. And we're able to ask how resilient to climate change are these sites at these microhabitat scales? Is there habitat management that we can do to improve refugia for these species under climate change? And where in the landscape should habitat be optimally restored? So we've been thinking about how climate interacts with seasonal light dynamics to affect lichen growth and performance, which are the most relevant climate variables in this temperate rainforest context, and vapor pressure deficit looks like a strong candidate. And then we're downscaling that to microhabitats to think about those microclimatic patterns. And my final example now is to try and place this information into a really applied conservation framework. And again, this is focused very much on these temperate rainforest epiphytes. They're globally rare. The UK has got high diversity and a high abundance of these species. So it's a great, it warrants this sort of conservation attention. Okay, but this is very much a thought experiment. So we want to think about what we can do to protect these species under climate change. And we want to do it in a really ecologically sound and integrative fashion. So we might think, right, we've got a wood. Maybe there's 1,000 trees in the wood, maybe there's 10,000 trees, but we can think of having a woodland. And then we want to know what is the amount of suitable habitat in that wood for these lichen epiphytes. So how many of these trees are likely to be suitable habitat where they can colonize and they can grow? So we did survey work across where we sampled lichen epiphytes across 1,000 quadrats. That was 250 trees at 20 sites. And we characterized their niche requirements. And tree species and tree size will go a long way to explaining which lichens you might find where. And there's an important difference for these temperate rainforest species between trees that have got a less acidic bark and trees that have got a more acidic bark. So you've got a higher likelihood of occurrence for trees that have got a less acidic bark than on a more acidic bark. And I just want to point out here, because this will be important in a couple of minutes, that those trees include ash and elm and aspen, some of the most important trees. But if we look at those niche models, then we can say, okay, well, in a typical woodland, let's just take a typical scenario. How much of the available habitat is suitable for colonization in a mixed deciduous broadleaf woodland on the west coast of Scotland? And it would appear to be about 80%. So we can say 80% of our trees are suitable and 20% aren't. And a fancy way of talking about that is to say the woodland's got a carrying capacity of 80%. And then in our thought experiment, we can colonize that woodland. So we can throw lichens out into the woodland to colonize onto the trees. And if they land on a suitable tree, then that's fine. They establish. And if they land on an unsuitable tree, then they don't. If they've landed on a suitable tree, then we want to know how long will it take them to reach reproductive maturity. So what's their generation time? Now we know this is again for in this case it's for Liberia pulmonaria, the lichen that we saw before, that as the thallus size increases, the probability of reproduction goes up. So there's a relationship between the size of the thallus and the probability that it's producing spores or asexual propagules. Now in continental Europe, it's been estimated that it takes about 20 to 35 years for Liberia pulmonaria to grow to a sufficient size to produce reproductive propagules. But when we do similar work on the west coast of Scotland, where there's a more optimal climate, then this growth phase 3, labeled here, occurs much earlier. This is a reproductive phase, and it's estimated that these lichens reach maturity after about 9 years rather than 30. So we can have a generation time of 9 years. We want some forest dynamics. So we want to think about what's the extinction rate for these species. And that could be, well, not the species, what's the extinction rate for individuals. So some individuals might fall off a tree or get knocked off a tree or blown off a tree. That's a stochastic extinction rate, or the tree might fall over. That's a deterministic extinction rate, but we're going to kill them off at a certain rate. And there are studies that we can borrow from that tell us what the typical extinction rate is in European mixed temperate forests, about 4.5%. So 4.5% per time step, 4.5% of our lichen individuals die, get rid of them. We'll regenerate the woodland to maintain the carrying capacity at 80%. So we'll put habitat, trees that have fallen over, we'll put them back in a way that maintains the same available niche space. And then we have a process of colonization. So our lichens can colonize over time steps. They can colonize from one tree to another, or they can colonize into our woodland from other woodlands in the landscape. So two different types of colonization. And we can characterize that because we did a study, one of our PhD students did a study where looking at an isolated tree in the landscape. And in this case, again, just to show, we have Laberia pulmonaria sitting on this tree. And then she had air samplers set out north, south, east, and west at different distances from the tree, sampling the air, so capturing propagules, and then she used DNA to look at the probability of the species having dispersed across certain distances. And then what you're able to do is characterize the number of trees that might be colonized over time for different distances here in the colors. So we have data on how these things might disperse in the landscape. But what we also need to do is to characterize the process of establishment as well. So you've got colonization and you've got establishment. So what's the required colonization rate to maintain the proportion of suitable habitat colonized? So although we've got the colonization rate, we need to know, for example, what the fecundity of the lichens is, how many propagules they're producing per unit time. And we can do that by thinking, in this case, about how much of that suitable habitat is actually colonized. So in a woodland, 80% of the habitat might be suitable, but we find that only 32% of that suitable habitat is occupied. So what we can do is we can run our models, we can run this thought experiment, this model over time. We can throw our lichens into this imaginary woodland, and then we can take these parameters, which, are guesstimates, but they're decent ones because they're based on data that we've got. And if we assume a probability of colonization of 0.118, then we will end up, as we run our model over time, with this 32% of suitable habitat colonized. So that seems to be our colonization rate that works, which is one colonization event per thallus every 8.5 years. Okay, so we've been through quite a, it's quite a simple process. We've got a simple set of parameters that we're using to model the population size of lichens in a typical woodland. Now, I hope this has been worth it, because now we can start to ask some questions, having been through all of that, and we can say, what happens? So we've got our population size here, we've got our model time sets, we've got all our parameters sorted out. We run our experiment, a sort of control burn-in up to a certain point, and then we can say what happens if we change the growth rate. So it gets wetter in the winter, there's less light, gets drier in the summer. When there's more light, growth rate changes. What happens if we decrease the growth rate and we increase that time to maturity from 9 years to 14 years? So it takes an extra 5 years to reach maturity. And in that case, then the population size drops. So the population size of the lichen will go down. And anything like that is going to be a problem, because this is a very simple model. We're just running a kind of equilibrium situation. But if we think about variable weather from year to year and so on, this line actually will wiggle around quite a lot. And the more you get, the closer you get to zero, the more likely it is that the population will just crash and it will be unviable. The next question, though, is what can we do to solve that potential problem? Changing climate, changing growth rates, falling population sizes. There are two things we can do. We can manage for more suitable habitat conditions, so for a higher carrying capacity, more suitable niche space in the woodland, or we can manage for more woodlands in the landscape, so we've got higher rates of colonization. So here's our drop in the lichen population And this is what happens if we manage for more woodland in the landscape. In order to reverse that trend, using these estimated values, but in order to reverse that trend, we have to increase the woodland in the landscape from about, this is native broadleaf woodland, from about 1.5% of the landscape today to 27%. So based on current planting targets, that's completely unrealistic. And it also takes a long, long time. If you implement that kind of regime now, it's going to take 200 years before you start to see the benefits, because that's how long it takes for these woodlands to mature and become suitable habitat. So the alternative is to increase the suitability of the habitat. And that turns around this problem faster. So having more suitable tree species of an appropriate age structure in the woodland, and you need to increase it from, say, 80% suitable habitat space to 94% suitable habitat space. So we need to be thinking about these kinds of solutions, and we need to be thinking about them fairly quickly. And it's reasonable to say, how are we doing in terms of increasing the suitability of our woodlands for these kinds of species? And I want to remind you that some of the most suitable species are elm and ash and aspen. And we've, of course, we've already had elm disease. We've got ash dieback, which if you travel up the west coast of Scotland is absolutely devastating populations of ash. And I don't want to get into the rights and wrongs of the reintroduction of the beaver. Very, very fraught issue, but we are seeing preferential gnawing and felling of trees like aspen, for example, particularly along watercourses. So whatever you think about that, the suitability of habitat at the moment seems to be going in the wrong direction. Okay, so on that kind of depressing note, some conclusions. So lichens, they're diverse, they're ecologically important for multiple different reasons. I think you can clearly argue that studying lichens, studying other fungi, invertebrates, microbes, it breaks down that taxonomic bias in science, and it does create, you are forced to think, think outside of those standard bioclimatic models. It creates a broader understanding of nature, and it helps reveal true complexity in the natural world. This isn't unique to lichens by any means, but it's emphasized by the study. Take climate change as an example. Getting down to the scale at which lichens grow, it reveals, for example, the important roles of species interactions, opens up the importance of different climate drivers when you're thinking about that biological knowledge, wind speeds, vapor pressure deficit, interactions with light, and how those are operating at these microclimatic scales to affect diversity. How are we doing to protect lichens against climate change? It's a really, really integrative challenge. You've got changing functional biology and growth rates, but those growth rates then have knock-on effects to population sizes that depend on how habitats are managed. So you've got this intersection of biology, population dynamics, habitat management. And I think the answer probably is at the moment not very well. Our ambitions are good, but probably habitat quality, particularly driven by ash dieback for these cyanobacterial lichens on the west coast of Scotland, is going in the wrong direction. And that is the end of my presentation. So just to flag, thank yous to funders. I'll say a special thank you to the British Lichen Society, who very generously provide a lot of data for this kind of work, and also collaborators who were involved in the work in this talk. So thanks, and I will stop sharing.
Well, thanks, Chris. That was fascinating, illuminating, and somewhat sobering all at the same time. And there's quite a bit of praise coming in as well on the chat there. If you bear with me one moment. So we'll break you in gently with the first question. I know we haven't got masses of time to do questions. The first one wasn't particularly lichen specific, but it was harking back to your initial summary of fluctuations of historical and projected CO2 levels. And you're being asked if you could give a brief explanation of what factors caused the historical decreases of atmospheric CO2.
Oh my gosh, that's a really difficult question. I don't know if I can't. No, I'm not going to go there. But it's a very it's a question that you can find the answer to by Googling it, but it's complicated because there's an interplay between the kind of planetary effects that shift temperatures on the Earth, and so the kind of Milankovitch cycles and so on, and then these kinds of feedbacks. So when the carbon dioxide levels in the atmosphere are changing over those glacial time periods, my understanding is that it's in relation to, it's coupled with those planetary changes, and it's created by a sort of a kind of it creates a sort of feedback. So as ecosystems draw down carbon dioxide, peatlands, seabed sediments, that kind of thing, and then as they release them again. Yeah. But the precise answer, I'm on thin ice there.
Okay, well, I read for one closer to the work that you've been doing. There were a couple of questions regarding your montane work, and could either the lack of competition from vascular plants or different plant communities in those montane habitats have effects on the densities or degrees of population of lichens in those areas?
Yeah, it could do. So when you shift, for example, onto the plateau, so you're going, these studies were done in the Cairngorms. So you're going up the side, I don't know if people know the Cairngorms, but you're going up the side of a mountain, and you know, you've got a mountain slope, it can be fairly even, and then all of a sudden you pop out onto a plateau. So you've got these plateau type mountains. And actually, yeah, the vegetation structure can be quite different. So it can be quite ericaceous as you're going up the side of the mountain. And then when you pop out onto the plateau, there's a lot of bare ground with sometimes with juncus and sedges that are forming these like spiky little clusters. So actually, the species are changing, but so is that structure of the vegetation. So indeed, I think that can be really important in why there are fewer of these mat-forming lichens on the plateau themselves. Yeah.
Okay, thanks. And still in the sort of same topic area, you've been asked if there have been any studies or if you've done any work on the wind speed effects in those environments, but on sort of crustose lichens that are on exposed rocks and things like that, or if you'd expect there to be similar effects.
I've not, no, but I would expect there to be a similar effect. And in the sense that I think some of those, that crustose diversity, that's really montane crustose diversity, is sometimes on the spurs of ridges where you get very, very high wind speeds and the snow is blown off. So that protective cover of winter snow goes. So really, really harsh environments. And it would be fascinating to look at the species composition and the diversity of those crustose lichen species in those kinds of habitats. Absolutely. Yeah.
Okay, brilliant. Now I know you have just picked up quite a few specific points, particularly towards the end of your talk there. but one of the questions had been, in response to climate change generally, what solutions could be implemented to protect lichen biodiversity? So is there anything else you would want to add beyond the points that you raised? I think the question had come up before you sort of half answered or answered that.
I think the point, probably the point I guess my general point, it's a good question. I suppose my general point is that climate change is important, but when we're thinking about the effects of climate change on lichens, it mediates itself through in other ways. So there can be a direct effect, but actually what it's doing is, like in the example I gave, maybe it's changing the growth rate and the time it takes to reach maturity, and that's got a knock-on effect on the population size, which we need to manage, through maybe thinking about habitat management, and how we manage our landscapes. So my general point would be that there is complexity, and that's because climate change isn't a factor operating by itself. It operates through an interaction with lots of other features of the environment that are affecting lichens. But the advantage of that is that it gives us tools to solve the problem, because it's this interactive network. Even though climate change might be happening, if we can get other things right, then perhaps we can reduce the risk by improving other features of the environment, creating larger populations, creating more resilient populations. Yeah.
Grand. There's lots of more questions coming in just now, but I don't think we'll have time for them all. One was referencing a recent paper by Nelson et al. That came out in Frontiers in Microbiology, that was suggesting that lichens' adaptive capabilities are too slow generally to enable them to cope with climate change, which fits a lot of things that you were saying. But do you have a particular view on that, if you happen to read the paper or were able to?
Yeah, I did see that. So I think it's a very provocative idea. But I think the jury is still out on that issue. So there is evidence for local population adaptation to different environments. So if you look at lichen populations across environmental gradients, there's evidence that they can be locally adapted. And then if you have gene flow between those populations, then perhaps there's the opportunity for adaptation to environmental change. But there's a huge amount of work to be done in that area, because there's a lot of uncertainty about whether the adaptive ability of lichens to climate change. They can also acclimate, so they can change their rates of respiration, they can change their investment in photobiont populations, and they can also change the photobionts, not like corals do by switching photobionts, the photosynthetic partner, out and bringing a new photosynthetic partner in, although there's some interesting work to investigate whether that can be done or not. But more typically through reproduction. So if you've got reproduction of the fungus, then there can be ecological selectivity for photobionts that provide a sort of optimum photosynthesis in a given environment. So there's also this photobiont switching that can be quite important as an acclimation mechanism to environmental change. Yeah. So I don't really have an answer for that either, except to say it's a really, really interesting area of work at the moment.
Okay, yeah. Grand. And that sort of leads in a little bit that several people had asked questions along the lines of sort of the fungal element, certainly of the lichens have got a sort of armory of chemicals that they've evolved to have, and how those different structures and chemicals may make them more or less adaptable in different situations. But I think you're just sort of saying there is the possibility for some to have surprise sort of levels of acclimatization or something, because they might have the ability to, you know, as you were just suggesting, switch.
Yeah, they do acclimate, and they can acclimate surprisingly quickly. So there are studies, for example, to show that the photosynthetic algae will acclimate across seasons, so their physiologies can shift. And then some of the chemicals, for example, are UV screening, so they sort of protect the fungus produces these chemicals to protect the alga from high levels of UV. And you can switch lichens between low light environments and high light environments, and the lichens will upregulate or downregulate that production of chemicals, for example. Yeah,
grand.
Yeah, so they're not always as kind of slow and passive as people think. They can be quite dynamic.
Yeah, fascinating. I say there were several people asking quite a number of questions along that. So I will round up. There's a lot of praise, a lot of particular comments directly about some areas of the work that you've done, and people are clearly fascinated. Something that has come up from quite a few folk was it's different questions along the same theme of either following the work that you're doing, or wondering, you know, sort of how they might be involved or be in touch or anything like that. So do you have any, apart from publishing in journals, is there any other way of people following the work that's being done?
So I'm not sort of very active on social media. We have our institutional website, and so it's worth going on there because there's little summaries of some of the work that we've been doing.
So that's through the Royal Botanic Gardens.
Yeah, exactly. Yeah, that's right. Yeah.
All righty. Okay, well, I say yeah, we've not time to actually go through all of the questions there, but it's been honestly fantastic talk. I'm looking forward to now listening to it again afterwards, where I can wholly concentrate on everything you're saying.
I don't listen. I'm not going to listen to it. I don't listen to it again. We'll see. Anyway.
Excellent. Well, thanks everyone that's joined us this evening, and thanks ever so much, Chris. It's been
you're very welcome.
informative.
No, it's been an honor. I was really delighted to be asked to speak here. Yeah.
Okay. Thanks very much. Enjoy the rest of your evening, everyone.
Thank you.
Bye.